Hello friends, we have already discussed about prokaryotic DNA replication in brief in separate video. We also discussed about DNA replication enzymes like helicase, primase, single strand binding protein and topoisomerases in detail. You can refer my videos, the link is given in the description box. Today we are going to discuss about prokaryotic DNA replication. We know that there are three events involved in DNA replication: initiation, elongation and termination. Here we will discuss about initiation. So before starting this, let us summarize what we have already discussed. First of all, what is DNA replication? It is the process by which cell copies its DNA or produce two identical copies of DNA prior to cell division. So before cell division, two identical copies of DNA is produced from the parental DNA, and then this is passed on to the daughter cells. This is important for inheritance, that is transfer of genetic material from one generation to another. There are certain aspects of DNA replication. First of all, DNA replication is semi-conservative. Semi-conservative means half conserved. It means in the DNA molecule, one is parental strand and the other is newly synthesized strand. So among the two strands of DNA, one is conserved and other is synthesized, which is complementary to the parental strand or the template strand. Also, DNA replication is bidirectional. When DNA opens up, two replication forks are formed at the origin. These are then extended bidirectionally. So let us understand it in this figure. So this is the origin of replication. Two strands of DNA separates. Replication bubble is formed and there are two replication fork, 1 and 2. And these are extended in opposite direction. This is extended in this direction and this is extended in this direction. So this type of the replication is bidirectional. That is, it proceeds in both the direction which are opposite to each other. DNA replication is semi-discontinuous. One is leading strand that is copied continuously and the other is lagging strand which is copied in the form of segments, that is known as Okazaki fragments. If we see here, this is the parental strand, this is leading strand which is synthesized continuously, and this is the lagging strand which is synthesized discontinuously in the form of segments. These are known as Okazaki fragments. Prokaryotic DNA replication is studied with respect to E. coli, which is model organism. All other prokaryotes show many similarities. So here we will discuss about prokaryotic DNA replication with respect to E. coli. In the DNA of prokaryotes, there is region where replication starts. This is known as origin of replication and these have specific nucleotide sequences. So if we talk about E. coli, this is the circular DNA of E. coli. This is the origin of replication known as ORIC, which is approximately 245 base pair. It has 3 copies of 13-mer region towards the left and 5 copies of 9-mer region towards the right. This 9-mer is DNA binding site. 13-mer region is AT rich region, which is the region where the two strands of the DNA separate or the melting of the two strands of the DNA takes place. It is AT rich region. AT base pairs are held together by 2 hydrogen bond and GC base pair are held together by 3 hydrogen bond. So AT base pair is weaker compared to GC base pair, and hence AT rich region can be separated or denatured easily compared to the GC rich region. Now there is a term known as replicon. It is a segment of the DNA that undergoes replication from single origin of replication. It has one origin and one terminus. In case of the prokaryotes, it has single replicon. Hence, prokaryotes are known as monorepliconic. There are three steps or sequence of DNA replication: initiation, elongation and termination. In this video, we will discuss about initiation of DNA replication in detail. So first of all, what is initiation? It is to start or initiate something. Sequence of events that takes place during initiation. First is recognition of origin. Origin is recognized by certain protein called initiator protein. In E. coli, it is DNA A. Now second is binding of DNA around an initiator protein complex, which consists of DNA A and ATP. DNA A protein binds to the 9-mer sequences, forming an initial complex, because 9-mer sequence is the binding site of DNA A protein. DNA coils around the protein. Now next is denaturation of 13-mer region. This coiling of DNA around the initiator protein, that is DNA A, induces topological stress. This causes denaturation of DNA at 13-mer site, which is AT-rich region. This we have already discussed. This is initial strand separation. That is, now the separation of the strand is initiated. ATP is required to form open complex, and for this strand separation, in order to form the open complex, energy is required in the form of ATP. Now next is loading of DNA helicase. Aggregate of DNA B, which is helicase, and DNA C, which is helicase loader. 6 molecules each of DNA B and DNA C is formed. So 6 molecules each of DNA B and DNA C forms aggregate. DNA C loads DNA B to the denatured region, and after loading of this DNA B, DNA C gets detached. So one molecule of DNA B, which is hexamer of identical subunits, clamp around each of the 2 single strands in the open complex. So in the open complex, there are 2 strands of the DNA, and DNA B, which is helicase, it clamps around each of the strand. For this, it requires energy in the form of ATP. Then helicase opens the helix. Further melting of the 2 strands to generate unpaired template strands. So helicase further helps in the separation of the 2 strands of the DNA, and 2 unpaired strands serves as the template for the synthesis of the new strand. Helicases move along a DNA duplex using the energy of ATP hydrolysis to separate the strands. So helicase moves along the DNA duplex, and it utilizes the energy of ATP hydrolysis in order to separate the strands, and to separate the strand, it breaks the hydrogen bond between the base pair. Then stabilization of single strand. Single strand binding protein binds to separated strands at the replication fork. So at the replication fork, when the 2 separated strands are generated, single strand binding protein binds it. This stabilizes single strand and inhibits from subsequently realigning. And because of the binding of the single strand binding protein, these single strands are stabilized, and these are not able to wind again and form the DNA duplex. They do not cover the nitrogenous bases, and these single strand binding protein are bind in such a way that they do not cover the nitrogenous bases. So these are easily available for the synthesis of the complementary strand. They hold them in a semi-extended position, hence are more accessible to DNA replication machinery. That is, these are easily available to the DNA replication machinery. After replication of particular segment of DNA, single strand binding protein molecules fall off and recycle to attach next segment of single strand. So once a particular segment of the DNA is replicated, then single strand binding protein falls off from that segment and then it attaches to the next segment of single strand. Then relieving of supercoiling takes place. Unwinding of the parental DNA by DNA helicases results in positive supercoiling. So, as the DNA helicase further separates the strands of the DNA, then supercoiling results ahead and behind the replication bubble. For the replication to proceed smoothly, it is important to remove the supercoiling, and this is relieved by topoisomerase, which we have already discussed in detail in topoisomerase video. You can refer it. So, this is relieved by topoisomerase 1 and topoisomerase 2. In E. coli, it is known as DNA gyrase, which is a type of topoisomerase 2. And supercoiling is relieved by transient single-stranded break. Then next is assembly of primosome. Primosome includes DNA G, which is primase, and DNA B, which is helicase. DNA B is required for the activation of the DNA G. So, after generation of replication fork, primosome assembles at the 2 fork. It initiates primer synthesis called priming because DNA polymerase 3 cannot initiate the synthesis of the DNA. It requires primer. And these primers are stretches of RNA or DNA that are synthesized by primase. In case of E. coli, it is RNA primer. And this priming or the synthesis of the primer takes place once for leading strand and repeatedly at intervals of 1000 to 2000 bases for lagging strand, because lagging strand is synthesized discontinuously, or it is not synthesized continuously. It is synthesized in the form of Okazaki fragments, and for each fragment primer synthesis is required. DNA G, which is primase, it synthesizes 15 to 50 bases of primers, and after primer is synthesized, primase dissociates from template. Once the primer is synthesized, then there is the initiation of DNA polymerization by DNA polymerase. And DNA polymerase results in the extension of the DNA strand or the synthesis of the DNA strand, which is complementary to the template strand. This we will discuss in detail in elongation video. Now let us understand the sequences or events of initiation with the help of the figure. So this is the supercoiled DNA of E. coli. This is the origin of replication known as ORIC. And this origin of replication consists of 3 copies of 13-mer region, which is AT rich region, and it is the region where the separation of the 2 strands of the DNA or melting of the DNA takes place. This consists of 5 copies of 9-mer region, which is the site for the binding of DNA A protein, that is the initiator protein. So this initiator protein binds with the 9-mer region. DNA wraps around this initiator protein, as a result of which stress develops. This results in the separation of the 2 strands of the DNA at the 13-mer region. For this, energy is required, which is used in the form of ATP. So as the 2 strands separate at the 13-mer region, it is known as the open complex. Now the further separation of the strand is required. This is done by DNA B helicase. DNA B helicase is loaded at the 2 replication fork with the help of the DNA C, which is known as the helicase loader. It also requires energy in the form of ATP. Once the DNA B is loaded, then DNA C, which is the helicase loader, is released. Now the further separation of the strands of the DNA takes place at the 2 replication fork in the opposite direction. Hence it is bidirectional. As the separation of the 2 strands takes place, single strand of the DNA is generated, which serves as the template for the synthesis of the new strand. In order to stabilize the single strand and to prevent it from re-annealing or rewinding, single strand binding protein comes and binds with these single strands. As the helicase proceeds further, or as the further separation of the DNA strands takes place by the helicase, unwrapping of the DNA takes place, which was wrapped around the DNA A protein. After this, priming takes place, that is the synthesis of the primer. For this, primosome is assembled at the two replication fork. This primosome consists of helicase, DNA B, and primase, DNA G. Helicase is required for the activation of the primase. So, primer is synthesized. So, these are the primer synthesized by the primase. This is the primer for the leading strand. This is the primer for the lagging strand. Here, this is the primer for the lagging strand and this is the primer for the leading strand. In case of the E. coli, it is RNA primer. After that, further replication takes place. DNA polymerase III comes and binds and results in the synthesis or the extension of the new strand of the DNA. This we will discuss in detail in the elongation video. So now let us see the proteins involved or required in initiation. First is DNA A, which is the chromosomal or DNA replication initiator protein. Second is DNA B, which is DNA helicase, which results in the separation of the two strands of the DNA. Third is DNA C, which is the helicase loader, and it loads the helicase to the two replication fork. Fourth is single strand binding protein. These binds with the single strand of the DNA and helps in the stabilization of the single strand and also prevent them from re-annealing or rewinding. Then fifth is DNA gyrase, which is topoisomerase, which relieves the supercoiling ahead and behind the replication bubble. So sixth is DNA G, which is primase, which synthesizes RNA primer in case of E. coli. And in this process, energy from ATP is required. It is required during melting of DNA by DNA A, which is initiator protein. Then the loading of DNA B, which is helicase, at the forks by DNA C, which is helicase loader. Then ATP is also required in the helicase action of DNA B, that is separation of the two strands of the DNA at the replication fork. Civil action of DNA gyrase. DNA gyrase, it is a type of topoisomerase and it relieves the supercoiling ahead and behind of replication bubble. For this process, it requires ATP. Then the activation of primase, DNA G by DNA B, which is helicase. Then activation of DNA polymerase III to begin replication. So ATP is also required for the activation of the DNA polymerase III, which begins the replication or which results in the synthesis of new strand of DNA. This is all for today's video. In the next video, we will discuss about elongation step of DNA replication in prokaryotes in detail. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.